Okay, so I would like to talk about an in vitro study. We combined radiotherapy with modulated electro-hyperthermia on a pancreatic adenocarcinoma cell line. Okay. So, little numbers about the pancreas carcinomas. The highest rate of pancreatic cancer is in Hungary. And is the 5th common case of death in Europe and the 7th in the Poland. But the most important number, I think, is this one. This is the 5-year survival rate according to the SEER. It doesn't reach 10%. This is because the poor efficacy of the actual treatments for this type of cancer. Why? Pancreas adenocarcinomas is really difficult to diagnose. There are no screening program for them, and the symptomatology of the disease it's very vague. No specific symptoms for this type of cancer. So the majority of the patients are diagnosed in advanced stages when the tumor is not possible to remove by surgery, and the remaining therapies are classical chemotherapy or radiotherapy or the combination of these two. So here can be the hyperthermia a possibility for these patients, and we use modulated electro-hyperthermia, which is a regional hyperthermia, and it uses electric fields to heat the malignant tissue to 42 degrees. So we treated the PANC1 pancreas adenocarcinoma cell line in this tank, filled with cell culture media, and we treated the cells for 1 hour. Yes, at 42 degrees. We had 4 groups: a control, hyperthermia treated, radio treated, and a combination of those 2 treatments. So firstly, we were looking to the morphological changes. We observed that in hyperthermia treated groups, those 2, appears apoptotic bodies and nuclear shrinkage. And after that, in 24 hours from the treatment, we counted the viable cells, and we found that significant less cells are viable after modulated electro-hyperthermia treatment and after the combination too. We measured the apoptotic and necrotic rate of those cells, and we found that this cell death is because of apoptotic cell death and not necrosis. Yes, the Cas+3 positive nuclei was significant higher in the combined treatment group, and also the H2AX positivity was the highest in the combination group. We were curious about the tumor progenitor cell number after the treatment. Those cells are responsible for high resistance of cells of tumor to the classical therapies. So we measured the HLD-A positive cells by flow cytometry, and we observed that after 24 hours from the treatment, there was a cell loss also in these cells which are HLD-A positive. The colony forming capacity also confirm these results. So our conclusion is that 1 hour of modulated electro-hyperthermia can lead to a massive apoptosis via Cas+3 pathway, And the combination of the true treatment can lead not only to a cell loss in the whole tumor, but also in the tumor stem cell culture. And that was. Thank you.
Thank you for your presentation. Are there any questions?
I got a little bit confused at one moment. You have still quite a lot of minutes left, but the slides went really fast. So at one moment you showed that you saw apoptotic bodies in the looking through your microscope,
Yes.
and you did an annexin 5 PI study, and they seemed to contradict each other, wasn't it?
No,
So the
I didn't.
annexin study showed a different profile in cell death and apoptosis than the apoptotic bodies, or not?
Yes, it was a higher number of apoptotic bodies on the charts too.
Because actually when I saw that, when I saw your cells laying there, I didn't see apoptotic bodies. I didn't see cell blabbing and that type of things.
They are. Sorry.
Or I was looking at it.
I was more with yellow. Can you please?
Yeah, so these yellow thingies. They
So here those pink cell remains on the slide.
yeah, these are cell remnants. I mean, I don't see cells typically coming off the background of their coating and then rounding off and then showing cell blabbing.
Most of the cells which are dying detached from the cover slip, so it is a lucky thing that we have a few of them here in the picture.
Okay, so then that's anoicus then.
Sorry?
That's anoicus then, that they detach and then they die.
Okay.
Because that I think we have to stop but there is a different mechanism there than that what you're saying. So you need to look at that.
Okay. Thank you.
Thank you all the speakers. Thank you my co-chair. And we will move to the awards session.